Hey, everybody, this is Jason Wilson with the Curious About Cannabis podcast. Thanks for joining us again. Today, I am joined with Dr. Kevin Spelman, one of my good friends, mentors, colleagues. Very excited to have you here today, Kevin.
It's a pleasure to be here, Jason. Thanks for having me.
Yeah, totally. Just to start us off a little bit, just to help our listeners understand who you are and the type of work you do. Usually, I tell people you're primarily a phytochemist. You understand a lot about plant chemistry, and as well as how compounds and plants interact with the body. Would you say that's a fair assessment?
I'd say that's a fair assessment. People tend to stick me in the chemist box a lot. I think of myself as a biologist because I'm fascinated by the way that plants affect our physiology, and I'm fascinated by that pharmacology. So by training, I'm a molecular biologist. But yeah, I definitely have spent a lot of time looking at molecules and looking at them in the lab and playing with them in the lab and looking at analytical chemistry results. And
Yeah, same here. That's very similar to me. I get pinned in the chemistry box these days because of all the analytical chemistry work I do. But at my heart, I'm a biologist and really more so an ecologist than anything else. But I think ecology translates really well to pharmacology in very interesting ways.
it's so true. And until there is this thin veil between them right now that keeps our lobbying efforts on more ecological and environmental standards from really being heard. Once the medical community joins the ecological community and the environmental community, we're going to have a much stronger voice. And that needs to happen soon because as we poison the planet, we're, of course, poisoning ourselves.
Right. And not to mention, just on a totally sort of anthropocentric perspective, losing potential novel compounds we haven't discovered yet. I think there's a lot of times a false perception that we already know a lot about nature and these compounds that are in plants and fungi and even in people, and that there's not a lot left to discover. And I try to explain to people that couldn't be further from the truth.
I completely agree with you. We are, I think as scientists, sometimes we're quite arrogant in what we think we know and forget about the dark knowledge that we can't see yet and that we don't know. A really good example of that, you brought up, losing compounds because we're losing plant species. We lose about a plant species a day.
Yeah.
And we've only researched about 6% of all the plant species on the planet. So what did we lose today, right? Was it a cure for Hodgkin's lymphoma or, HIV or, yeah. So we've got a lot of work to do. And I want to say the number of estimated plant species is something around 500,000. That's a fuzzy number for me, but 6% of those is all we've done.
Yeah. There's a long, long way to go.
A long
for
ways
sure.
to go.
Yeah, totally. To jump into the cannabis side of things, do you mind describing? I know you've done some research in the past around the endocannabinoid system, and a lot of work, which I want to spend a lot of time talking to you about today, about plant synergies and compounds in other plants other than cannabis that interact with the endocannabinoid system. I know you've done a lot of work with echinacea.
Yep.
And so do you mind just talking a little bit about some of that research that you've done and kind of see where that conversation goes?
Sure. Well, let's start with the endocannabinoid system. I did have an opportunity to look at some signaling going on there, CB1 and CB2. That's not to say that the endocannabinoid system is only CB1 and CB2. That's, I think, a misunderstanding that is frequently, what's the right word here? That's a misunderstanding that is frequently used to talk about an endocannabinoid system. But the endocannabinoid system is really fascinating. I look at it as a lipid signaling network that if we were to diagrammatically look at it, I would say that it's at the bottom of a pyramid, and everything else, neurological system, cardiovascular system, immunological system, all of those are stacked on top of it, and therefore that foundation endocannabinoid system affects everything that's sitting on top of it. So it has this huge role in signaling throughout the body, and I think that's really, really key. And so it's so much more than just the enzymes that are synthesizing
Hmm.
and breaking down cannabinoids and the receptors that are directly considered cannabinoid receptors such as CB1, CB2. Now there's a putative CB3.
Hmm.
It looks like there's probably a CB4 and a CB5 at this point, but it's too early to actually call them that. But there are a lot of compounds there are a lot of receptors that actually bind our endocannabinoids as well as the exogenous
Hmm.
cannabinoids in our body.
Yeah, and are you referring to like a lot of those G protein coupled receptors?
Yeah, GPCRs, exactly.
Yeah.
And then you've got the ion channels that are coupled to receptors. So the whole TRP receptor system is really fascinating and makes a huge difference in terms of pain and sensation, as well as, just gut health
Hmm.
and neurological health. And so yeah, it's a very pleiotropic system that affects so many different subsystems or tissues throughout the body.
Yeah, yeah. It's funny, the more research that goes into the endocannabinoid system and looking at these G protein coupled receptors that endocannabinoids interact with and everything, it's just the definition's getting wider and wider and wider. And while a lot of cannabis education is still on that, okay, the endocannabinoid system is CB1, CB2, these enzymes, these ligands, which for anyone listening that doesn't understand what the word ligand means, that's just any compound that interacts with another receptor. We call those ligands, kind of simplified definition of that. But it is important for people to understand the endocannabinoid system is very broad, and the TRPV1, the TRIP V1, and all the other TRPV ion channels, those are starting to get investigated. TRIP V1 is now pretty much considered an intimate part of the endocannabinoid system in a lot of newer papers that are coming out, but it is much wider. It's tricky because when you're talking to a lay audience that isn't familiar with a lot of
Yeah.
medical science and all of that, you kind of have to keep it simple. And how would you describe molecular signaling to someone that is sort of new to trying to understand what the endocannabinoid system is, what a receptor is, what a ligand is, all these sort of things? How would you describe that system? Because normally it gets described as like a lock and key model, which we now know is not appropriate.
Yeah, thanks for saying that. I'm so tired of hearing about the lock and key model, and we're hearing it from a lot of marketing now in the cannabis industry. But the lock and key model, that old model was there was the one key fits one lock, and that could be nothing could be further from the truth in terms of that. We're finding that receptors tend to be promiscuous, if you will. Especially, I spent a lot of time looking at these receptors, orphan nuclear receptors.
Mm.
Inside the nucleus, the PPAR receptors, PPAR gamma is so incredibly promiscuous that two different molecules, two different ligands can fit in there at the same time.
Oh wow, I didn't know that.
Yeah,
Wow.
it's pretty fascinating. So, and that's by the way, is also considered part of the
Hmm.
endocannabinoid system now. Is the PPARs, these orphan nuclear receptors, and they're called orphans because the original ligands were the the endogenous ligands were never really established. There's definitely some deep insights into what they are or could be, but when you hear that orphan, it means that we don't really know what the endogenous compound that relates to that receptor
Mm.
is. So, yeah, I think we have. I agree with you completely. It just keeps getting broader and broader, and it's gone well beyond CB1, CB2, Fa, MAGL. Those enzymes, and it's an exciting time to think about it in terms of signaling. Coming back there, um, how does signaling happen? Carefully, number one, it is very um, orchestrated in a very profound way that I don't think most people realize. And so that first piece there that it's orchestrated, I want to refer to this idea of It's a network. And this really comes from a lot of ecology understanding, where we look at ecosystems and we understand that they're very complicated networks. You can't just introduce rabbits into it, right? Or whatever it was.
Or pull something out.
Yeah, you can't pull something out or put a species in. In the same sort of way, you can't block a enzyme or a receptor system without having profound implications on the rest of the network. So genes, receptors, enzymes, all these targets, as we call them in pharmacology, respond to messenger molecules. Is a term that I really love because it makes it really clear what these molecules do. Each molecule carries a message, and so these molecules usually bind more than one site, and that's something that's misunderstood as well. And and by the way, then their metabolites, they're broken down by enzymes, and then those metabolites of those molecules then can bind other sites. So we have this incredible network that I like to think of it as network pharmacology. There's this model now that I think is really starting to embrace medicinal plants in a way that couldn't be embraced when we were looking at one target, one ligand model. So signaling is incredibly complex. It's orchestrated. It's about harmony. It's about all these signals happening simultaneously and sequentially, and therefore having a response that makes sense to the body. And without that complex network signaling that includes upregulating and downregulating genes, includes upregulating and downregulating receptors, includes upregulating and downregulating enzyme systems. Without all of that, we would be puddles of slime on the floor.
Right. Yeah. I mean, literally, it's you. If that intimate, I like the orchestration model, this sort of orchestra or symphony of activity that's going on. If things don't come together, you can't even think straight or perceive properly. There's a number of different things besides just like bodily functions not working. Well, an example I've talked about before, but I think illustrates the sensitivity of messing with these systems is, did you hear about the study that was done in France a few years ago, where they tried to do it was an FAH blocker.
Inhibitor,
It ended up sending several people to the hospital. One guy brain dead, went majorly wrong. And there's been some follow-up studies to try to figure out why it went wrong, because the rodent models made it seem like it should have been no problem, which I don't know if that's something you want to comment on at all. Because there's a lot of cannabis research currently. A lot is either cell culture studies that are looking at how cannabinoids are interacting with certain tissues or cell types, or different proteins like receptors, or they're rodent models, and results are getting scaled. And particularly this issue has come up just in the past couple of days. Forbes released an article about CBD, and the headline said something like, researchers find CBD causes liver damage. Very broad title. And I was like, oh, that's interesting. And I wondered, are they talking about the cytochrome P450 stuff? What are they referencing? And so I went and looked up the study, and, it was a rodent study, and they ended up taking their lowest dose of CBD that they gave the rodents was allometrically scaled based on the highest doses that went through the Epidiolex trials. So looking at 15 to 20 milligrams per kilogram body weight, scaling that down to rodents, that was their starting dose. And then they did doses that were 3 times and 10 times that. And what they found is in the doses that were 3 times and 10 times, they saw some pretty significant liver damage that started to occur in the rodents, particularly in the 10X model. They were feeding these rodents 2 grams of CBD per kilogram of body weight.
Holy cow.
Yeah, over 2 grams.
And then a journalist blows it up into, oh my God, my hair's on fire.
Right, yeah. And it's something that myself and several other people on social media that try to do science education, and particularly around cannabis, tried to actually post the article and say, no, actually read the research study, because the authors even talk about, something that you and I have talked about before, super physiological doses.
Yeah.
And that while these effects occur, in most real life scenarios, you're unlikely to encounter them because most people aren't consuming that much. But is that something you can comment on at all? Just some of the nuances that people should be aware of when they're looking at studies that are using rodents versus humans, how that scaling occurs, and
Yeah, so let me tell you a story that shocked me. I was running a R&D group that was a company. It was $1 billion company, so they had money and they had resources. In fact, my research budget was $10 million. And there had been a nutritionist there for many years that apparently was doing the dosing on any of the they did, this company did multivitamins, so he would figure out the dosing. And so when I asked him, he mentioned a study to me, and I said, "What is the HED, human equivalent dose?" And he gave me a blank look. Now, he had been there for over 10 years determining this company's dosing. And I said, "Human equivalent dose? Do you know what that is?" He said, "No, I've never heard of that." So he had been determining all of his dosing based on animal models. And of course, the human equivalent dosing models are based on either surface area or metabolic rate. And hopefully they're based on both, and that'll give you a more accurate read of what the right dose might be for a human in an animal model. But he had never heard of this, and so I sent him all these papers on human equivalent dose that I had on my hard drive, and he never mentioned it again because I think he was incredibly embarrassed that he'd been determining doses based on animal studies that were not relevant to humans. So there's the first piece, is like people tend to, if it was 2 grams a kilogram, they tend to just multiply that times 65 or 7. Actually, the average human now is over 65, at least in the U.S., kilograms. So they tend to do that, and that's wrong. Metabolically, I think a mouse its metabolic rate is 7 times faster than a human. I think rats are 11 times, and then you've got surface area to deal with as well. So those doses tend to change pretty significantly. But coming back to the animal model, I think we have to be really careful when we're looking at doses. Basically, anything over about 50 milligrams per kilogram is probably not realistic for human use. It's on that cusp, and that's even super high. So you get up to something like 2 grams, I mean, you're toxic. And that would happen with, look, you can drink too much water and drown, right?
Right.
So anything can be toxic, and that kind of brings us to this idea of hormesis and understanding what hormesis is. And this is that idea of a biphasic response, that a little does one thing, but a lot does another thing. And generally with this hormetic response, a little bit of a substance that potentially could be dangerous actually upregulates things like NRF2 or NRF2, it's called, which then launches this whole antioxidant system, protective buffer system. But then if you have a lot of it, it's toxic. And this is seen throughout all kingdoms, whether it's plants, whether it's animals, whether it's reptiles, whatever it is. And it's there were in the 40s, actually in the 40s is when it started losing favor. But throughout the early 20th century, there were journals based on this hormesis model. It wasn't always called hormesis, but there were a number of names for it. So we need to understand that. So the American way that if one is good is not two better, anything worth doing is worth overdoing, right?
Right.
That's not true, and we need to be really careful about that in medicine, in food, in supplements, that a little bit can go a long ways, and when you take more, it doesn't mean you're getting more of the same effect. It may mean that you've gone to the other side of the curve, and you're actually getting potentially toxic effects.
Yeah, and this is something that in medicinal plant science has been well known, that there are a lot of different plants that may be helpful, but if you're consuming a lot of them every day, it's known that you start to get liver problems, all sorts of different things. Even I was talking to someone recently just about reishi mushroom tinctures, and just explaining like, hey, just understand that like this isn't something you just consume a lot of all the time and you're going to be super healthy. Like you've got to understand the dosaging, understand what you're working with, periodically take some breaks, let your body kind of reset, and then pick it up again, and give your liver a break from having to process, some of these kind of foreign compounds, that sort
I
of thing.
completely agree. The idea of drug holidays is should be applied to dietary supplements as well.
Yeah.
The one thing I would say though, in terms of dosing in dietary supplements, especially on tinctures, look, you got a 1 ounce bottle,
Right.
and it's often now over $15. I've seen them at $25. Oh, but in the cannabis industry, they're 4 times that.
Right. Yeah. Like $150 for an ounce.
Yeah, right. But the doses they have on those bottles usually are subclinical.
Yeah.
And so, for example, with echinacea, 30 drops is not going to do it, right? We're talking teaspoons, if on the first signs of a cold. But I completely agree with you. The idea we need to think of dietary supplements as food items, which is really
Yes.
in many ways what they are, and therefore we wouldn't eat nothing but blueberries for 5 days, right, straight. We would have judicious amounts in our diet, and that's how we need to think of dietary supplements. So, one of the things that I think isoflavones is a really good example where the isoflavones from beans, and usually people just think soy is the only place, but isoflavones are in basically almost all legumes,
Yeah.
that these compounds actually bind the beta estrogen receptor, and it's at a much lower affinity, but they still have a slight estrogen nudge towards the body. So, in our diets, maybe 50 milligrams a day was what you could get if you were eating a lot, if you were a Japanese or an Asian person eating a lot of soy-based foods. But I see supplements with 150 times that, or excuse me, 150 milligrams, rather than just 50 milligrams. So I worry about that sort of thing.
Yeah. Yeah. And it's weird. Like now, culturally, microdosing is sort of in, but in the 90s and early 2000s, megadosing was really in, and especially around like vitamin C.
Yeah.
That was huge, taking mega doses of supplements to try to get certain responses that now a lot of research around that mega dosing is not upholding some of the initial thoughts of what people thought that might do. Well, talking about these other plants, let's talk about plant synergy and herbal
Sure.
synergy and the endocannabinoid system and how that all comes together. I think probably a lot of people listening to this have probably heard of the entourage effect. That's become really popular in the cannabis space. And a lot of people act like that's a new thing.
Thanks for saying that.
But I think, as you can attest to, it is definitely not a new thing, and something we've been trying to understand for a long time. And cannabis is kind of bringing that to the forefront. So can you just kind of describe how you conceptualize these herbal synergies, entourage effects, whatever you want to call them, and then I'd like to get into talking about things like the alkylamides in echinacea and other compounds, some of the terpenes that are found in basically every plant,
Right.
that are interacting with the endocannabinoid system, and I don't know how people should think about this, because it's getting so much hype.
Well, I think you used the word symphony. That's really what I think the best depiction of this idea of the entourage effect, or what we have called for probably longer synergy in medicinal plants, is a symphonic effect. So think about a Mozart symphony that's so incredibly beautiful, and you have a lead violin, and then you have all these other instruments, right? And all those other instruments basically support that lead violin. Now, what's happened in pharmacology is, under the illusion, and I will call it an illusion, that we can control the dose, we've isolated the lead violin and just used that in our pharmacology. And the rationalization for this is actually, if you really go back and look at the science, the rationalization for doing this is actually not very strong scientifically. In the 30s, in the early 20th century, 20s, you had the eclectic physicians doing studies on whole plant extracts, and then in some cases they actually used an isolated constituent. And this is the Lloyd brothers. The eclectic physicians were supported by some brilliant pharmacists called the Lloyd brothers, and the Lloyd brothers were really good at extracting. In fact, some of their instrumentation, their chemical instruments are still used today. They invented them to do extractions, and we still use them today in chemistry labs. So they had this opportunity to look at whole plant extracts versus isolated constituents. And they found in many cases, not always, but they found in many cases that the whole plant extracts were had a better outcome. Now, and I think that's really what we have to look at, because in an isolated model, for example, a cell culture in an animal model, you actually might get a better outcome with an isolated constituent.
Right.
Because you're pounding, essentially it's pharmacological dosing versus physiological dosing, right? So in physiological dosing being that idea of using just enough to get the effect you're looking for.
Minimum effective dose. Yeah.
Exactly. And what we end up seeing with this in healthcare is that everybody's too busy to do that. The patient's too busy to pay attention, because they're going to have to pay attention to where they get the effect. The doctor's too busy to actually put an inquiry into where was that dose that worked for you. And everybody wants instant relief. So it's fallen by the wayside. But were we to pursue that further, I think we'd have a much more effective model. The other thing that happens here when we look at isolates versus whole plant extracts is the adverse event profile goes up. And this is a huge issue. And for example, in my own body, if I take ginkgo 24/6, the standardized stuff, I actually end up with a huge diuretic effect. I'm running to the bathroom every 20 minutes. If I just use ginkgo leaf that's thrown in a capsule, I don't have that effect.
Yeah.
I notice the blood flow effect in terms of cognitive function with both of them, but I'd rather use the whole leaf ground up. So we have that lead violin that's working. And what we found in reductionist chemistry now is that, there's more than one lead violin. Turns out that the oboe takes a lead in this section as well, right? And it turns out that the cello also ends up having a lead in the. So we have found multiple active constituents. In the case of echinacea, it's been hypothesized that at least four now compounds are considered the actives. When they came across the CB2 binding of alkylamides, what happened is everybody thought, we've finally done it. We have found the active in echinacea. Well, not so fast, because we've got the caffeic acid derivatives, which, by the way, are directly antiviral, but incredible antioxidants and potentially block histamine release. By the way, if you're looking at anaphylactic reactions and you're stuck in a pinch. We've also got the polysaccharides, the rabinogalactans in echinacea that are also super active and directly stimulate immune function. So that becomes very interesting, because here in one plant you've got something that's a bit of a immuno dampening effect, the alkylamides, and you've also got something that's a direct immune stimulator, that's the rabinogalactans, and then you've got something that tends to modulate immune function, that's a caffeic acid derivatives. And by the way, it's antiviral. So we can't simplify plants to one constituent, and that's the mistake that's being made over and over and over. It started in the 70s with ginseng. People were looking at ginseng and saying, what's the active constituent? And they came upon the ginsenosides, or the saponins of the plant. And so what happened from this ridiculous research in the 70s is there was a time when people would look at plants, and if they didn't have alkaloids or they didn't have saponins, they were thrown out. No, they're not active.
Yeah.
It's all fantasy. No, these tribes using this are not getting any effect. We know best.
Right. We've studied it.
Exactly. So I could talk about this all day. I've written a couple of chapters on it in books, and published a couple of papers on synergy. But the idea that there's only one active compound is really naive when it comes to medicinal plants. And we need to look at it as there's this symphonic effect when every time we swallow a bolus of either a food plant or a medicinal plant, we're getting literally thousands of compounds, and I literally do mean thousands.
Yeah.
The idea that there's only 400 compounds in cannabis, I think, is super naive as well, because if you look at corn and you look at wheat, respectively, they have found 3,000 and 5,000 different compounds in there, and these are probably the most studied plants, right, analytically. Yeah. And so I can tell you with certain clarity that there's at least 1,000 compounds in probably every single plant. As analytical chemistry gets more and more
widens,
sensitive, it's just going to widen further. So to think that there's only one active compound is delusional. And I think we need to step back and look at this network pharmacology idea where we have multiple actives, basically nudging a network, not blocking any one site 100%, or not binding with great inhibition or
Right.
great agonist activity.
Partial agonism, partial antagonism.
Exactly. Partial inhibition. And that combined effect of multiple nodes in a network being nudged has potentially a better outcome and a safer adverse event profile as well.
Well, there was one study just looking at CBD. I believe it was with CBD, looking at isolated CBD, and then I think they call it a whole plant extract, which the way that some of these terms are used in the industry drives me crazy.
Yeah.
Because, in herbalism, the term whole plant extract does have a definition. You're using aerial parts, you're using roots, all these different pieces. If it's an extract just from aerial parts, you say that, but anyway, beyond that, there was a study that showed that with isolated CBD, there was a very limited therapeutic response profile, and it's pretty much a pretty standard bell curve.
Yeah.
And that you, at certain dosages, you hit a peak, and then you start getting losses in therapeutic efficacy. But when they used these more, for lack of a better term, broader spectrum extracts, or whatever extract they used, that curve turned into a linear line, that they started to see better therapeutic outcomes with fewer side effects and better outcomes at lower dosages. But the main thing is that ceiling was seemingly eliminated when they introduced an extract versus an isolated compound. And I can't remember exactly how they measured all of that. I need to go back and pull it, but that's just...
It was a TNF alpha study.
Was it? Okay.
Yeah.
Yeah. And so that just in a very narrow way demonstrates the huge difference between isolated compounds and extracts that have a broader spectrum of phytochemistry. And it's tricky. I want to know your thoughts on, so the traditional model of doing clinical trials and all of this is, standardized ingredients that are the same every time, very tight controls. What's the way forward given that medical science, and I do think this is becoming a more and more popular notion now, the idea of network pharmacology, understanding that these limitations of isolated compounds, the more and more people I talk to, people seem to be accepting that. But the pushback that I receive is, but how do you study it to get that included in medicine as our systems are set up right now? Because it's very hard to get funding to do clinical trials with herbs because there are much greater limitations into what product you're gonna come out with and market.
Yeah.
And so, what's the way forward to do research to identify the therapeutic potentials and the limitations of medicinal plants, given this model that we're stuck with?
Yeah, it's a really good question, and I'm going to give it a really simple answer, but the devil is in the details. And the answer is outcomes versus mechanisms. What we're looking for a lot of times in clinical trials is, of course, we're looking at outcomes, but we really want to understand mechanisms in a way that may not be realistic. And I was actually just at a traumatic brain injury conference, and I suggested the idea that there's dark pharmacology. They were talking about dark data that was negative, that never got published. I turned it on its head and said, let's talk about dark pharmacology. Dark pharmacology is pharmacology we can't see yet because we've been looking under the street light, right? Looking for the drunk that's looking for his keys, right? That's what we've been doing in science. We look for what we know and not necessarily, and by the way, it's expensive to go off into the weeds. Looking for something. So we start thinking we know everything. And I've seen this happen with a lot of medicinal plant studies where, St. John's wort doesn't work for depression because it doesn't do this and it doesn't do that, the mechanisms that we know about. Well, actually, if you look at the outcomes, it actually does work. And so I think one of the answers is really, when we can't necessarily standardize the amount of active compound A or active compound B because they're medicinal plants, we have to look at the outcomes. And I think that's really the key. And there's been a lot of they're starting to happen. Let's not say that there's been a lot of them, but there's more and more of these studies on integrative medicine, complementary alternative medicine, whatever you want to call it these days. I just call it medicine myself. But
yeah,
there's been these studies basically looking at seeing alternative practitioners who do multiple things, right? It's not just one drug or one herb. They're suggesting this, and I want you to think about that, and I want you to read this book, and I want you to take these 3 herbal compounds, and one's a tea, and one's a tincture, and one's a capsule, and they're all doing different things. And then, that confounds the way that we've done research. And I think the key is outcomes and not necessarily mechanisms. The mechanisms are sexy, and believe me, as a molecular biologist, I'm seduced by molecular modes of activity, right? I want to understand, but it also gives us a false arrogance.
Yes.
That we think we know, and in fact, there's so much dark pharmacology right now that we don't know, and we only know what we know, and we do not know what we do not know.
Right. Yeah. There's a massive amount of humility that needs to be had by anyone working professionally in medical science and really science broadly. I mean, science has repeatedly been plagued by arrogance, a false sense of that understanding of like, oh, I know enough now that now I can run with this and extrapolate to, the ends of the universe. And
in the early 1900s, they wanted to close the patent office, right?
Right, yeah. It's like, no.
The steam engine has been discovered.
Right, we're good now. And no, I think that's a really good point. And to add to that, when I get into discussions about this with other people, I talk about assigning value to the safety profiles of a lot of these medicinal plants like cannabis, and recognizing that, we should feel more confident doing that type of research focused on outcomes given the safety profile of these plants and these compounds. Because if nothing else, we have a pretty good sense of how toxic a lot of these things are, at least when it comes to LD50s, lethal dosages, and that sort of thing.
And let me interrupt you there, right there, because I think you just brought up a really key point that I can hear the scientists, not so much, and the medical doctors probably very much saying, safety? What are you talking about? Let's talk about safety for a second here, because I'm going to try not to go off on a rant here.
Go for it if you want to.
But you end up with somewhere between 80,000 to 100,000 people die every year from properly prescribed pharmaceutical medications. And if we have 5 deaths in a year from dietary supplements, it's blown up all over the news.
Oh, yeah.
And if you look at the numbers, more people are using dietary supplements than are using pharmacological medications or pharmaceutical medications. Right. So we need to step back and go, this is not a safety issue. It's an economics issue.
Yeah.
We've got the pharma crying foul because we don't have to do the amount of research from substances that are naturally occurring, that most of them have been in our food, in our diet anyway. So, they're saying, well, they don't have to do this and that. Well, that's true. And maybe that is something to look at. But to say that they're not safe, I don't think so.
Yeah, no, I totally agree. I think that's a bullshit reason to limit research and encouraging people to
fear their dietary supplements.
Yeah. Well, and to touch on something you said earlier, talking about dietary supplements, thinking about them as foods. Foods are medicines, and it's important for people to recognize that and understand, like, the things you eat, they are chemicals. They're influencing your body. They're triggering all sorts of signaling in the body. Your foods are medicines as well. Pharmaceuticals aren't the only medicines, or standardized supplements and pharmaceuticals are not the only medicines. It's everything you consume is a medicine, and it's important for people to think critically about what they're consuming broadly.
Absolutely. There's a phrase that I'm going to attribute to Jeff Bland that I actually really like, and that is that you steep your genes daily in your dietary broth.
Oh, that's great.
Isn't it?
Yeah.
And if we looked at a fast food eater versus somebody who just, had their greens and some fruit and maybe some nice protein, whether it be vegetable or animal-based protein. And we looked at the gene profiles, what's been upregulated and what's been downregulated. And those two people, they would be completely different. And suddenly you would understand why that fast food eater is going to die 10 years earlier than the other guy is.
Right.
But we don't do that.
Right. Yeah. Well, it's fascinating to me that all of the education that goes on, commercials that come on TV, of health campaigns and everything, it's such a simple message that for some reason isn't coming through, that like, hey, your food is also medicine, and, trying to encourage that holistic thinking about health and wellness. And even in the concept of dietary supplements, a lot of people, especially in the United States, fall into the same sort of thinking of like, oh, I'm sick, I need to take a bunch of supplements, treating it just like they would a pharmaceutical, and not thinking of like, well, what's my daily lifestyle? What's my daily regimen? And how do I incorporate things into my daily routines to support my body as it is? And a lot of people aren't even doing, going back to these, like taking breaks from things, like trying to figure out what your baseline is so that you even know what you're working from,
Right.
To know what you might need to incorporate.
Yeah, most people don't know that.
Yeah, no, it's very challenging. And I mean, I'm as guilty as anyone else, so I want to make that clear. Like, I'm not perfect. I eat plenty of fast food and all sorts of, but it's about developing that awareness and trying to make your decisions be more mindful, and just having your whole life be more mindful and trying to be in touch of what you're feeling, what you're experiencing, what's influencing you broadly. And that extends beyond just food, but everything, news and media, all sorts of things. What's influencing you? What's building your reality? Because it's all of that. All of that itself is a symphony, an orchestra.
Exactly.
That can be very discordant if you're not taking that time.
Yeah, you bring up two really good issues here: industry and lifestyle. The industry, the food industry, does not want to know about those studies and has actively tried to block the release of such studies in terms of showing that a healthier diet has a very profound influence on upregulation of positive genes and downregulation of negative genes, the negative effects, versus fast food. So we have a huge lobbying effort to prevent that sort of information getting out and actually doing those studies. Let's take the Harvard researchers that hid the sugar research and pushed the high fat research, right, to protect an industry. And you got lifestyle. And, having lived in Europe and having lived in Latin America, I have to say, Americans, we're freaking crazy.
Yeah.
I mean, the way we work, and I'm at the top of the heap in terms of being a bad, lifestyle guy, and just the amount of work that I do in any given day, and then I don't stop. I usually work my weekends off as well. Life is really super busy, but that is not the way to be a healthy person. You've got to have integration time. You've got to have downtime. Our brains can't it's like, you're going to run your engine on high all day long. You wouldn't do that to your car, right? But we expect it from our bodies and from our minds. And so there's a lifestyle mismatch with what is healthy, or we think we're going to get rewarded for busting our asses. And in fact, what we're really doing is degrading our health.
Yeah, no, completely. And sabotaging the very thing that you want, because you generally want to be high performing, you want to perform well and all these things. And by stretching yourself too thin, you're actually making your performance worse and making it more likely that you're going to experience negative outcomes later in whatever.
Yeah.
So
back to cannabis.
Yeah. So I love it though, because what I like about cannabis is it's a doorway for a lot of people to get into these very critical topics. And that's why this podcast, I don't want to limit it to only talking about cannabis growing and extraction, all these things. While I do want to talk about those things, I want to get into the deeper concepts that cannabis is sort of a springboard for. But that being said, coming back really directly to cannabis, what do you think are some of the biggest misconceptions that people have either about cannabis or the endocannabinoid system that you tend to encounter on a regular basis?
There's a couple, and I would say that they're both actually what I like to call black market hangovers. One is the idea that if we're going to use THC medicinally, that the requirement is that we get high from it.
Really good point. Yes.
And I'd have to say nothing is further from the truth. THC is pharmacologically active at doses that don't generate a high or euphoria. And there may be a couple instances in severe pain or cancer where you really are dealing with the central nervous system effects of THC. Right. But otherwise, there's many cases where that's just completely not necessary. And then the other one that just is driving me crazy, and it's definitely a black market hangover, is decarboxylation. This idea that we have to blow off CO2, that we have to...
Got to activate.
We got to activate, right. And, this has been shown. In fact, I've just compiled a list, working on a white paper right now, and I've just compiled a list of all the targets of acidic cannabinoids. And let me tell you, it's big, especially for CBDA. It's huge. I have a friend that was at UCSF for years, a family nurse practitioner who handled kids with intractable seizures. And what she did was made sure that the cannabis oil, the hemp oil that they were getting, was non-decarboxylated.
Yeah.
So she was using strictly CBDA with these kids and getting great results.
Yeah.
And so, and also TRPV1, which by the way, CBDA binds at high affinity, is a target for seizures. So it turns out that CBD is still working, but CBDA works really well too. So we do not have to decarboxylate everything. And there's probably advantages to not decarboxylating in terms of bioavailability as well.
Yeah, and man, that first point you brought up has been on my mind a lot lately, that you should not equate therapeutic response with the high. Yeah, just the higher you are doesn't mean you're getting more therapeutic response. And I think that's something that just takes people a while to catch on to, especially if they're really new to cannabis, because that's the main entry point, is that they know that cannabis gets you high and it helps people, and so I'm going to experience a high if I want to use cannabis therapeutically. That's really, really important for people to understand. There are so many things happening in your body that you're unaware of, that you just can't perceive directly, and that's why it's really, really important to try to take that time to inventory yourself. I talked about it before. I'll talk about it probably every episode, but journaling when you're trying to treat yourself with any therapeutic agent, taking really careful notes about dosaging, when you're taking things, what you're noticing day to day, even just really basic things. Monitor your pain levels if you're trying to treat pain on 1 to 10, and monitor that,
Encouraging self-awareness.
Yeah, just trying to really critically evaluate your state of being, because a lot of these activities, other than the sort of euphoric effect of THC, most of these effects you're not going to feel, not right away. Some in pain relief, if it's immediate joint aches or whatever, you might notice that. But a lot of other stuff, it's something that builds over time as well with repeated dosaging, especially with cannabinoids or any lipophilic therapeutic agent. You're going to have that building up over time in your body. And so, I think I'm really happy to hear you talk about that. I think that's really important for people to recognize. And I'm sure there are some THC producers out there right now that are unhappy to hear that.
Cursing at the radio or whatever their device is.
I've already warned people that this podcast, it's going to get me hate from every angle. And that's how I assume I'm doing something right. I'm prepared for that. So that's fine.
I just have to credit Ethan Russo. I like the way Ethan says it about THC and the high, the euphoria. He says that if you're using THC, cannabis, chemovar, THC, therapeutically, and you're high, you're having an adverse event.
Right. I mean, in any trials that are done with THC-rich cannabis, that is one of the adverse events that is monitored. And the only time I could see that being different is certain psychological conditions
Yeah.
where, the high may be part of the therapeutic response. But other than that, no. I mean, unless you're using it recreationally.
Yeah.
In which case,
Fine.
that's fine. Yeah, that's your prerogative to do. But don't confound the recreation aspect of cannabis with the therapeutic aspect, because they aren't always married together, although sometimes they are.
Yeah.
So what do you hope to see as cannabis research continues, and it's picking up at a really fast rate if you're monitoring the papers that are being published? I mean, basically since last year, there's just been a huge increase in momentum.
Half a dozen to a dozen every week.
Yeah, it's really wild. It's hard to keep up with.
Yeah, it's a full-time job.
It really is. It's something that, I try to connect people with research papers as best I can and stay up on top of things as best I can. But, I'm in a perpetual state of being behind, and I'm realizing that's just the way it's going to be because, all this research is coming out. It's like I'm going to have to rely on really well-written review papers at this point to stay caught up. But what would you like to see from the future of cannabis research and endocannabinoid research?
A couple things. One thing that is super fascinating to me is looking at the new research on frontal striatal connectivity in the brain with CBD, with cannabidiol, which, so what I basically just said is synaptic patterning is strengthened or even potentially becomes more complex. And I think that's super exciting. We're talking now about not only protecting the brain, but we're also talking about making it more agile in a sense.
Right.
And so that's super fascinating to me. But the other piece, and I think more basic, Greg Gerdeman is the scientist who discovered the retrograde signaling of cannabinoids.
Yeah.
And Greg basically just says it beautifully. He says something to the effect that there is a mountain of research showing neuroprotective effects of cannabinoids. And yet we have a scientific community that until very recently has been based on the negative effects of cannabinoids. And so, sure, any medicine, anything that's going to be medicine, whether it's a dietary supplement or a pharmaceutical drug, has potentially, it's a double-edged sword, has potentially a negative side.
Yeah.
But the neuroprotective effects of cannabinoids are striking. So I'd like to see more acknowledgement of that from the scientific community.
Yeah. Yeah, I agree. And broadly, man, I just want to see more healthcare professionals get educated on some of this stuff.
That's a really good point.
Because, man,
it's needed.
It's, it's very hard to find a doctor or even a nurse that understands much of this in any sophisticated way. And a lot of those that do operate in the cannabis space still don't have that in-depth of a understanding. I've been pretty disappointed by different folks that I've talked to that are actively doing work with patients with cannabis that are still talking about indica and sativa as if it's a significant model to use for finding different effects that things will have, or even talking about strains and not recognizing that you can have different chemotypes among things that are labeled the same strain name. Cultivar names can be helpful in getting you pointed in the right direction towards certain things, but if you're just telling patients to use a certain strain of something to go to their dispensary and find some X strain to treat X condition, X, Y condition, like
you don't know what they're going to come back with.
No, and that's just not really an appropriate way forward. So I don't know. I hope things get caught up quickly because our healthcare system needs it, I think, to really push this critical thinking about cannabis and, getting back to the springboard effect. I think it will help healthcare professionals broadly start to open their minds a little bit about medicinal plants just across the board and I hope to see that humility piece get cultivated.
Yeah, you bring up the humility piece is huge, and I unfortunately don't see that changing, but I hope I'm wrong.
Yeah.
I lost what you just said before that. Cannabis, what was it?
I was saying, hoping they have a better understanding, and that springboards into broader acceptance of medicinal plants.
Thanks. Yeah, that was a piece I was going to comment on. Yeah, so in regard to medicinal plants, I think that we need to realize that cannabis is just one medicinal plant, and everybody's excited about that medicinal plant right now. But that's my hope as well, is that this idea that a medicinal plant that's a complex array of phytochemistry and phytonutrients has the potential to make a very meaningful therapeutic difference in the quality of people's lives. That, to me, is medicinal plants, and that's for 30 years. That's what I've been focusing on, is medicinal plants. And I've made enemies in scientific institutions merely because of what I'm into,
Yeah.
and not because of my science, but because of the fact that I have an interest in medicinal plants. There is this bias in science against medicinal plants, as if it's not real. And we need to stop and realize that about half of all of our drugs have been based on medicinal plants, and about 25% of all of our pharmacopoeia is still directly isolated from medicinal plants. If you look at cancer drugs, something like 70% of them in the last 10 years have come from natural products in some sort of way. So when physicians and healthcare providers look at me as, with a disapproving look that I'm into medicinal plants, they don't understand their own pharmacopoeia that they're writing from daily.
Yeah. No, I totally see that too. Well, and so you've been extremely generous with your time. I'm going to go ahead and start to wrap this up. One of the last things that I wanted to ask you is, leaving cannabis aside for a second, what's at least one thing that you're super excited about that has nothing to do with cannabis?
I'm super excited about lion's mane right now. I've had an opportunity to look at some of that research, and recently wrote a review paper on it. And the hericinones and the erinacines, these diterpene derivatives that aren't polysaccharides, that aren't beta-glucans. Everybody's excited about mushrooms, and everybody thinks it's all about the beta-glucans. It's not. They seem to have a pretty profound effect on things like Alzheimer's disease and mild cognitive impairment. And so I'm super interested in seeing that therapeutically harnessed to make a huge difference in the quality of aging people's lives.
Yeah. Lion's mane mushroom. I've got some dried out lion's mane in the cupboard now. Something I'm very fascinated as well. Well, thank you so much, Kevin, for coming on and talking to me. We'll have to sit down again sometime and pick up discussion. I want to keep track of your work and what you have going on, and I'm sure we'll have plenty to talk about again soon.
I'm sure. Jason, thank you. I love what you're doing here, and getting the word out. It's great. Thanks.
Thank you very much. All right. If you want to learn more about Curious About Cannabis, you can go to cacpodcast.com or check out the Curious About Cannabis book, available now on Amazon and other online retailers. Thanks, and have a great day. Take it easy.